This essay example, as well as thousands of others, is available in text format on our website for free and without registration. Simply Google Ivy Panda Free Essays.
Introduction. Photosynthesis is the process by which plants assemble carbon-based compounds, which are the building blocks and energy stores of life. Plants first entrap sunlight energy and convert it to chemical energy in ATP molecules, which are in the form of bonds. ATP brings energy to reactions where glucose is formed from water and carbon dioxide. To finish, glucose molecules are combined to form starch and other molecules. Oxygen is also produced during photosynthesis and released into the atmosphere. The process of photosynthesis is summarized in the equation below. 12H2O plus 6CO2 equals 6O2 plus C6H12O6 plus 6H2O. Aerobic respiration is a procedure of cellular respiration that utilizes oxygen to split molecules to release electrons and form energy. In this process, adenosine triphosphate, ATP, is produced, which is liable for storing up and transporting most energy to other body cells. Aerobic respiration has two byproducts, water and carbon dioxide. It usually involves three main stages of reactions: glycolysis, which includes Krebs cycle, and electron transport phosphorylation. The equation below is a summary of aerobic respiration. C6H12O6 plus 6O2 equals 6CO2 plus 6H2O. How the two processes are linked between plants and animals based on the reactants and products of both pathways. The two processes are the lifeblood of plants and animals. These processes link in the way that the byproducts of one process are used as the raw materials of the other. Photosynthesis uses carbon dioxide and water from aerobic respiration to produce oxygen, food, glucose, and water. Aerobic respiration in animals will require glucose and oxygen from photosynthesis to produce energy, ATP molecules, and carbon dioxide and water will be used again in photosynthesis. A description of how energy is transferred from sunlight to ATP, from ATP to sugars, and from sugars to your cells. Sunlight is trapped by organelles called chloroplasts in the form of chlorophyll, red and blue light, to start the process of photosynthesis. In this process, molecules of carbon dioxide, gas, and water are combined in the presence of solar energy, and chemical energy is formed. Calvin's cycle then takes place to convert ATP to sugars through carbon fixation, where 6 molecules of carbon dioxide are combined with ribulose biphosphate to form phosphoglycerate, PGA. It is then converted into G3P, glyceraldehyde 3-phosphate, which is a sugar. The sugars are then consumed by human beings in the form of starch. The role of fermentation in allowing an organism to generate energy for its cells in the absence of oxygen. In the deficiency of oxygen, pyruvic acid can be converted into compounds such as lactic acid through the combination of glycolysis and other additional pathways in the process of fermentation. This is important during exercise, especially because breathing cannot provide the body with all the oxygen needed for aerobic respiration, and the cells turn to lactic acid fermentation, therefore providing the muscles with the energy required in exercise. How the energy from the sun ends up as chemical energy for the anaerobic organism or cell. Before fermentation occurs, one glucose molecule is split into 2 pyruvate molecules through glycolysis, summarized as C6H12O6 plus 2ADP plus 2P plus 2NAD plus equals 2CH3CoCo plus 2ATP plus 2NADH plus 2H2O plus 2H plus. Thereafter, fermentation can take place where sugars are converted into cellular energy, producing carbon dioxide and ethanol because of the absence of oxygen, as shown below. C12H22O11 plus H2O plus invertase equals 2C6H12O6. C6H12O6 plus zymase equals 2C2H5OH plus 2CO2. How an enzyme catalyzes a reaction. During a reaction, a substrate that requires processing is carried towards the enzymes. Enzymes accelerate reactions by lowering the free energy of the activation barrier, which is the EA barrier. The enzymes are substrate definite and, therefore, can just speed up the creation of one form of a substrate. Usually, weak hydrogen or ionic bonds join the substrate to the enzyme. Then, the enzyme lessens the EA barrier of a reaction by appropriately adjusting the substrates, damaging substrate bonds, and giving a good microenvironment for the reaction to occur in the optimum pH temperature and, i.e., and participating thoroughly in the reaction. There are three main steps in the cycle of enzyme-substrate interactions. Enzyme plus substrate. Enzyme-substrate complex. Enzyme plus product. How is enzyme activity regulated by the cell? Cells regulate enzyme activity through end product inhibition. The enzyme catalyzing one of the stages in the metabolic pathway is inhibited by the end product. Subsequently, if the quantity of product swells, the pathway is hindered and less is formed. If the quantity reduces, the inhibition is condensed and more is manufactured. Additionally, the gene that produces the enzyme is possibly switched on or off by courier molecules, for instance hormones. This essay on biology, photosynthesis and respiration was written and submitted by a student just like you. The IVPanda samples database hosts thousands of works similar to this one, on various subjects. Visit ivpanda.com to find what you are looking for, free of charge.